to mute your phone, mute your Zoom for now, that would be fantastic. And I'll just kind of take a quick second to introduce myself and talk a little bit about what we're going to go through here today. So my name's Dr. Steve Friedenberg. I'm a geneticist and a veterinarian at the University of Minnesota. I'm also like a criticalist, so I treat animals on the clinic floor, in the ER and the ICU. And I have like a strong clinical interest in studying autoimmune diseases in dogs and sort of complex genetic traits, and have been working to try to understand the genetic basis of Addison's disease for kind of years now. It's a pretty challenging problem. And I started sort of looking at this disease in standard poodles, and as of the past couple of years have been also looking at it in Portuguese water dogs. And I wanted to take this opportunity to talk a little bit about Addison's disease in general, and then also talk a little bit about my research and some of the very early findings we have from the Portuguese water dog work, and then kind of go from there. The plan is for this seminar to take about an hour. I'd like it to be kind of informal. There's only about 30 people on the call, and so what I'll do is, after kind of the main sections of the talk, I will give people an opportunity to answer and ask any questions, and I'll do my best to answer them. You can, I do have the chat open. So during that time, you can conceivably ask questions in the chat, or you could use the kind of raise your hand function in Zoom, and I'll call on you then. Hopefully it'll sort of be able to see you in the screen here. So lastly, I'll sort of say this is being recorded. I think we're going to post this somewhere. Janet Boyd will take care of posting it, excuse me, on the Portuguese Water Dog Foundation website. And so you can sort of have other people take a look at the recording, or people can sort of email me with questions later. Okay. So all of that said, then I'm going to kind of go ahead and get started. And let me click on the right screen. Okay, yeah. So here's what I'm going to talk about today, just a little bit of an overview of the talk. So we'll talk a little bit about what we understand the pathophysiology of Addison's disease to be. We'll then kind of get into trying to understand a little bit about the clinical presentation, so how these dogs look when they get sick, how we treat dogs with Addison's disease. And then probably the last half to third of the presentation, we'll talk a little bit about my research, which focuses, even though I'm trained as a geneticist, a little bit beyond genetics, looking at both the immunology of the disease and trying to understand what kind of factors in the environment can also be associated with Addison's disease. One of the things I'll kind of try to stress over and over as we talk about the pathophysiology of the disease is Addison's is not really likely to be sort of just solely a genetic trait. There's probably interfaces between the environment and genetics that kind of all makes this disease happen. So anyway, that's kind of where we're going to go through. So let's start off by talking a little bit about the pathophysiology of Addison's disease. So Addison's disease, as many of you guys know, is a disorder of the adrenal gland. And I'm hoping that my pointer shows up here. I may sort of change this to, let me see if I can change this to a laser pointer. So hopefully, does this show up here in red with this? Yes, can you guys see that? Mary, can you shake your head if you can see the red? Okay, it looks like yes, great. So Addison's disease is an order of the adrenal glands. And so the adrenal glands are these like triangular shaped organs. And this is a picture of the adrenal glands in a human, but in the dog, they looked a little bit the same. There's sort of these triangular shape on one side and peanut shaped on the other side, organs that live in a human just on top of the kidneys. And in a dog, everything is kind of rotated 90 degrees. So the adrenal glands live kind of right in front of the kidneys. And they're these small kind of inconspicuous organs. A lot of times they're even hard to find when we look at them. This is an image from a dog of what a right adrenal gland looks like on ultrasound. It's kind of this peanut-shaped organ. Here it's kind of encased in fat that lives in the retroperitoneal space, which is really where anatomically the kidneys and the adrenal glands are located. Um, and one of the things that you can maybe start to see when you take a look at this peanut-shaped organ is there's this kind of section in the middle, right? There's this line here that I'm tracing that actually separates two parts of the adrenal gland. So the adrenal gland is really kind of a pretty complex organ. And the next slide, which I'm going to show you here, kind of zooms in on that in sort of what we might see if we were to look at the adrenal gland under a microscope. And so when you look at the adrenal gland under a microscope, it's really broken up into 2 or 3 main sections, right? And the first section is the capsule, and a lot of organs are surrounded by a capsule. It's just a fibrous tissue that contains the organ. It's not very active. But the 2 main active parts of the organ are this outer part called the adrenal cortex, and this inner part called the adrenal medulla. And kind of interestingly, when you think embryologically, or the way that the adrenal gland is formed, the adrenal medulla and the adrenal cortex are kind of totally separate organs. So the adrenal medulla is the organ that sort of is essentially part of your nervous system. It's the part of the body that releases epinephrine. So you think about the typical fight or flight response that we can all experience when we're nervous, or when we're being chased by a tiger or an elephant or something like that. And so the nervous system will tell the adrenal medulla to secrete epinephrine. And that part of the adrenal gland is really totally and completely unrelated to anything that we think about with Addison's disease. What's important when we think about Addison's disease is this outer layer of the adrenal gland, which is called the adrenal cortex. And the adrenal cortex itself is even itself complex when you look at it under a microscope. You can begin to see that within the adrenal cortex, there's actually 3 different layers of the adrenal cortex. And this outer layer is called the zona glomerulosa. This middle layer is called the zona fasciculata, and the inner layer is called the zona reticularis. It's not obviously important that you remember those names, but it does sort of ultimately relate to the pathophysiology of Addison's disease, because each of these different layers of the adrenal cortex secretes different hormones that are important for life, and that really don't get secreted when we have patients with Addison's disease. So what happens in Addison's disease is, we believe that there is an immune-mediated destruction of the outer layer of the adrenal gland, again, the adrenal cortex. And what we think happens, I'm going to sort of show you on the next page here, is that this is actually a picture from a dog with Addison's disease, is that the adrenal gland somehow becomes infiltrated with all of these kinds of cells. And the cells here are immune cells. They're called lymphocytes and plasma cells and macrophages. And it's all of these kind of conglomeration of blue dots in the middle of the screen. So these cells should normally not be there. They should normally be kind of some immune cells in the adrenal gland, but not big clusters of them like you see under the microscope here. And what you're seeing is you're actually seeing these immune cells destroy the adrenal gland of a dog that has Addison's disease. This just happens to be a dog that, for whatever reason, passed away in the throes of kind of Addison's disease, and we're able to kind of catch the immune cells actually destroying the adrenal gland when you look at it under a microscope. If a dog had been fully sort of in the end stages of Addison's disease, the whole adrenal cortex is actually completely obliterated. So just kind of going back to a slide here that I showed you before, all of this tissue, the adrenal cortex, would really just be replaced by fat at the end stages of Addison's disease that would totally be not functional. And you would just look and it would go right from capsule, a little bit of fat, and you would go right into the adrenal medulla. The adrenal medulla is totally and completely spared in dogs with Addison's disease. Okay, so now why is it important to understand a little bit about this pathophysiology? So, just getting back to what the adrenal gland looks like under a microscope, you have here on the left kind of a cross section of the adrenal gland where you can see the cortex is this pink stuff here and the medulla in the middle. And if you were to blow it up I mentioned this earlier, there's really these three layers of the adrenal gland, the zona glomerulosa, fasciculata, and reticularis. And you can kind of see these different types of cells, right? The cells here look a little bit different than the cells here, which look a little bit different from the cells here. And each of these different layers of the adrenal gland secretes different types of hormones. And these hormones are essential for life. And the outer layer of the adrenal gland secretes this hormone called aldosterone, which is a really important hormone. The middle layer of the adrenal gland secretes this hormone called cortisol. And the inner layer of the adrenal gland secretes certain hormones, which are called sex hormones. But clinically, the most important hormones that are important in the pathophysiology of Addison's disease in the dog are the ones that are secreted by the outer two layers of the adrenal cortex, the zona glomerulosa and the zona fasciculata. And those hormones, again, are aldosterone and cortisol. And because in Addison's disease, we at least believe that the immune system destroys the entire adrenal cortex, without these cells there to produce these hormones, the hormones go away. And now all of a sudden you have a dog, or a healthy dog is sort of walking around with plenty of aldosterone and cortisol that are secreted during the times of the day when they should be secreted, whereas a dog with Addison's disease is going to suffer from the absence of both of these hormones, aldosterone and cortisol. Okay. And so what do these hormones do, right? It's important to understand the role of these hormones, because understanding the role of these hormones, what they do in health, we can think about what happens when you don't have those hormones is really what determines what we see in dogs with Addison's disease. So normally cortisol, there's a lot that's been written about it on the web, and people think of it primarily as like the stress hormone, and that's totally correct. So cortisol is a hormone that is increased by the body whenever the body is under any kind of stressful situation. So for me or you, that could mean sort of like we're about to give a talk or a presentation in front of a large room of people where it's something we don't like to do. For a dog, it could be going to the vet. It could be going to a new doggy daycare, whatever it is. There could be lots of things that, being pregnant, right? All those kinds of things are stressful situations and could require increased amounts of cortisol to be secreted by the body. In addition to kind of being the stress hormone, there's a number of other things that cortisol does. So for example, cortisol will tell the liver to produce glucose, which is sugar. Cortisol will also counteract the effect of insulin. So insulin is a hormone that you may have heard about because it's important in diabetes. Insulin's important for the body cells to take up glucose. And so it would lower our glucose, and cortisol normally counteracts that effect. There's also anti-inflammatory effects that cortisol has. So it can sort of suppress the certain transcription factors, which are proteins in the body that tell the DNA to make protein that are really important for inflammation. It has an important role on regulation of the blood vessels. The smooth muscle of the blood vessels are sort of the muscles that help determine whether they're going to be dilated or narrow. Cortisol has effects on fat metabolism, bone formation, and also acid secretion in the stomach. But in Addison's disease, it's really these couple of effects here that I've highlighted in red that are really important. So because cortisol is important in stimulating glucose production in the liver and counteracting the effects of insulin, oftentimes without cortisol, dogs tend to become very hypoglycemic, which means their blood sugar can drop to kind of dangerously low levels. And that's something that we oftentimes see in dogs with Addison's disease. We oftentimes don't see, because the anti-inflammatory effects of cortisol, so dogs can have, can be a little bit inflammatory when you look at their blood counts on a CBC. And then also there can be sort of trouble because you have trouble in maintaining the smooth muscles of the blood vessels, particularly dogs that are in an Addisonian crisis can be very difficult to resuscitate because their blood vessels don't constrict in the way that a blood vessel should when a dog gets very sick. So all of those effects can really be important to think about in Addison's disease. When we switch over to cortisol, the other hormone, aldosterone, what does aldosterone do? So again, aldosterone, another important hormone that gets lost in Addison's disease. And here I just have a little cartoon where we're going to start here on the right, where you see aldosterone. It's this sort of green circle here. So aldosterone gets secreted normally into the bloodstream. It goes into a cell called the principal cell of a kidney. And what it does is it goes into the nucleus, or the inside of the center of the cell, and it tells the DNA of that cell to make these important channels in the kidney. And one of those channels is important for taking up sodium, and another one of those channels is important for getting rid of potassium. And so this is normally what aldosterone does, is it tells the body to take up sodium and get rid of potassium. And what we see in Addison's disease, if you don't have aldosterone, we see exactly the reverse of this. So most of the time, dogs with Addison's disease, because they can't take up sodium, the sodium gets excreted into the urine and it becomes really low. And because we can't get rid of potassium, potassium builds up in the body and potassium gets really, really high. So when you look at it in total, without these hormones, what do we actually see? So you have a number of different effects that are primarily mediated by this hormone called aldosterone. And we see, for example, low sodium, high potassium. We also see lots of fluid loss and dehydration, because as the body loses sodium, it can also lose water into the kidney. This excessive fluid loss and dehydration, in its worst cases, can oftentimes lead to a shock state, where dogs don't have enough blood volume for what they need to deliver oxygen and nutrients to the tissues. And we can also see important changes in the pH of the blood, which is important for normal enzymes to work, secondary to the shock state. Because we lack cortisol, we see a lot of these effects that I've highlighted here in blue. So we see low blood sugar from impaired liver function, difficulty in maintaining the integrity of blood vessels, which is important to counteract the shock state. So we have sort of a double whammy. And we also see the absence of a stress response because of the lack of that cortisol. So that's kind of really all I'm going to say as far as the underlying pathophysiology of the disease. Is going to kind of move on to talking a little bit about the clinical presentation, but I'll just take a second to see if anyone has any questions about the pathophysiology of the disease before we go ahead. And feel free to put it in the chat or just raise your hand. Okay. So not seeing any questions pop up quickly, I'm going to kind of move on to the clinical presentation of what we see in our patients. So in terms of the clinical presentation, dogs with Addison's disease really present in one of two different ways. And the first way that dogs can present is in what's called an Addisonian crisis. And I would say somewhere between about a third, maybe 20 to 30% of dogs will present in the state of an Addisonian crisis. And this is one of those things that we want to avoid, but sometimes, either the Addison's disease can develop very quickly, or for whatever reason, people can miss what's going on in their dog and just miss some other clinical signs, which admittedly can be very, very vague in dogs. And the dog will sort of get sick enough that it presents in an Addisonian crisis. And so what do we mean by an Addisonian crisis? So this is a cardiovascular state of shock, where because of the lack of aldosterone in the body, so much water has been lost through the kidneys. There have been things like vomiting and diarrhea that's contributed to more fluid losses. And also we have a high buildup of potassium in the blood. And all of that, those significant electrolyte abnormalities can be imminently life-threatening. And so whenever, dogs come into an emergency room in these kind of shock states, veterinarians will evaluate the dog. You can evaluate whether you can see whether or not a dog is in a shock state by looking at their mucous membranes, by feeling their pulses, by checking the temperature of their limbs, by looking at something called a capillary refill time. Oftentimes, when a dog presents an emergency, we'll run some routine blood work that will tell us things about very, very high potassium levels. And when potassium levels get really high, when a dog comes in this kind of a shock state, they're really at an imminent risk of death. And a veterinarian needs to intervene immediately with fluid therapy and other treatments that we would use in a dog with Addison's disease, for example, to lower that potassium to a non-life-threatening level as quickly as possible. And so, again, about 20 to 30-ish% of dogs will present in this Addisonian crisis. The rest of the dogs will present with Addison's disease, what I would just call as like a routine presentation. And so Addison's disease in dogs, is oftentimes referred to as, quote, the great pretender, because the symptoms that we see in dogs with Addison's disease can really mimic those of many, many other conditions that we see in veterinary medicine. So my dog is lethargic. My dog is a picky eater, right? That term hyporexia means they have a poor appetite. My dog has kind of some intermittent diarrhea, or maybe she vomits every once in a while, right? Where these signs come and go, but they so overlap almost, anything. You can sort of look at a dog wrong and the dog will like decide to vomit for whatever reason. And so it's one of those things that we see. They're very, very non-specific, and so it can be hard to know what's going on. Now, I think most veterinarians are pretty clued in to the fact that Portuguese water dogs, standard poodles, a number of other breeds have a very high incidence of Addison's disease. So if you think a dog that's presenting with these kind of vague signs, a lot of times we'll do some routine testing to try to rule in or rule out Addison's disease very early on. But particularly in breeds where the disease is not so highly predisposed, right? Because we see Addison's disease in, for example, mixed breed dogs. Okay, great, thank you. Because we see Addison's disease in like mixed breed dogs, it can be a little bit harder to pick up. So there is this kind of, again, vague and routine clinical presentation, but it is how many dogs are in fact diagnosed. And we really want to prevent dogs from getting to this other stage. The other thing to know about Addison's disease in dogs, which is actually a little bit unique to dogs, is that there are really two types of Addison's disease that we recognize in dogs. And one kind is called typical Addison's disease. And in typical Addison's disease, there's an absence of both glucocorticoids with your cortisol and mineralocorticoids, which is the hormone aldosterone. This is a disease that's typically diagnosable on routine blood work. Because we don't see sodium, we don't have aldosterone, we typically see very abnormal ratios of sodium to potassium that we would see on really routine blood work. But dogs with typical Addison's are really the dogs that are prone to the more severe clinical presentation of that Addisonian crisis, because the absence of aldosterone leads to excess fluid loss and leads to an excess buildup of potassium in the bloodstream. So those dogs tend to be the one that have a more severe clinical presentation. Treating typical Addison's disease, because we lose both hormones, treating dogs with Addison because you lose both cortisol and aldosterone requires treatment with multiple hormones, not just one, okay? And I would say probably about 80 to 90% of dogs have typical Addison's disease. There's another form of Addison's disease called atypical Addison's disease, which leads to an absence of glucocorticoids or cortisol only. It's really pretty hard to diagnose on routine blood work, like a CBC and a chemistry profile that most veterinarians would run, because you don't see these big changes in sodium and potassium. There might be some subtle changes that you would see on blood work, like you, for example, could see some subtle abnormalities in the white blood cell count, or you could see, like, for example, a slightly low blood sugar. But the clinical presentation is a lot more vague. The advantage of having atypical Addison's disease, once it's diagnosed, is it only requires one medication for therapy, and we'll talk a little bit about treatment in just a little bit. But sometimes this atypical Addison state can progress to typical Addison's, but we just not all the time. So sometimes dogs with atypical sort of stay at the atypical state. Okay. Now, the other thing when we think about the clinical presentation for Addison's disease, it's really important for you guys to know, and I think everyone knows this, and this is really why it's here, is that Addison's disease is highly overrepresented in several dog breeds. And there's been a number of different studies that have come out about this. I always cite this study, which is from the Journal of Veterinary Internal Medicine from 2015, just because it's overwhelmingly the largest. And many of you guys may know, but in Sweden, many dogs have health insurance. I think maybe almost all dogs are required to have health insurance. And so you can kind of go through insurance databases of claims. And this group from Sweden sort of did this, and they looked at a population of 525,000 plus dogs to try to understand which breeds were overrepresented for Addison's disease relative to other breeds. And when you sort of look at these numbers, right, the breeds that have a statistically significant increase in the incidence of Addison's disease, you can see that Portuguese water dogs are absolutely at the top. And, this is the Swedish population. Could it be a little bit different in the US population or the Australian population? Potentially. But I think, we all sort of recognize in Portuguese water dogs and standard poodles, these are kind of the poster child breeds that they teach us in vet school to look for, to know about for Addison's disease. And when you sort of look at the numbers, there's this number here that I'm highlighting with my cursor called relative risk. And so what this means is, what is the risk of developing Addison's disease in each of these breeds compared to an average dog in Sweden in this study? And you can see that Portuguese water dogs are 29 times more likely to develop Addison's disease than an average dog. Standard poodles are 17 times more likely, bearded collies 7 times, Cairn terriers 3 times, Great Danes 3 times, Cocker Spaniels about 3 times as well. And so, there are some other breeds on this list that sort of don't make the cutoff for statistical significance, but this is just sort of the data that we have, probably the best epidemiologic study looking at the incidence of Addison's disease across the world. And it looks like there's a comment here, surprised not to see boxers in the frequency list. Probably if you were to look way farther down on the list, I've just kind of cut it off for the sake of display. You'd see like boxers and German shepherds and things like this. These aren't all the breeds that were studied, but these are just sort of what I've cut off for the sake of presentation. Okay. Now, I'm going to sort of, the other sort of really fascinating thing when you sort of get into the genetics of this, and, I've shown this slide before, but this is this is sort of something in scientific terms we call a cladogram. I like to sort of refer to it in a little bit of a tongue-in-cheek way. This is the wheel of dog relatedness. And I realize it's very, very hard to look at see exactly what this is showing, but if you sort of take a step back and just look at the big broad circle, what this is showing all of these dog breeds. I think there's roughly 170 or something like that represented on this figure. And it's meant to show the relatedness of each dog breed to the next breed. And this is based upon genetic data called the SNP arrays that were sort of put together by Elaine Ostrander and Heidi Parker and their colleagues at the National Institutes of Health. And the closer that a dog breed is to the other one, the more highly related they are. And so you can sort of see groups of dogs here, right? Like all of the light green dogs and the blue dogs and the light blue dogs are all highly related. But the thing that I just want to point out to you here is when you look at this slide, if you look in where I have this first circle up at just about the 12 o'clock position, is where I've highlighted standard poodles and Portuguese water dogs. And you can see that those are the 2 breeds that are most highly related to each other across the entire dog world. And lo and behold, Portuguese water dogs and standard poodles are the 2 breeds that are most highly predisposed to developing Addison's disease. So what does this suggest to us? It suggests to us that there must be some sort of a shared genetic predisposition or inheritance pattern that kind of is shared between Portuguese water dogs and standard poodles. Do we know that for sure? Absolutely not, right? It's certainly possible that Addison's disease evolved separately in Portuguese water dogs and standard poodles, but your hypothesis, a good hypothesis, would be that it kind of is shared just because the dogs are so closely related. Just sort of as an example for you guys about an unrelated disease, and I didn't create a circle here, but if you look at where my cursor is now around the Shetland sheepdog and the collie, so those dogs are very highly related to each other. They're the closest related dogs to each other, and they both share a common genetic inheritance for an autoimmune disease called dermatomyositis. And that's a disease that one of my collaborators who's worked on this project as well worked out the genetic inheritance for. And it's sort of the same principle. Highly related breeds, highly likely to share a common genetic inheritance. Interestingly, though, when you think about Addison's disease, some of the other breeds on the list were the bearded collie and the cocker spaniels. And those breeds live at very, very different positions on this wheel of dog relatedness. I've sort of highlighted them. The bearded collie lives down here at about 6 o'clock, and the cocker spaniels live over here on the left at about 8 o'clock. And you would question whether the inheritance, the reason, the genetic basis for Addison's disease is the same in all of those breeds. We don't really know. We don't know much about the genetic inheritance of Addison's disease in any dog breed. But if you had to guess, if you wanted to sort of start with your low-hanging fruit, you sort of might think about looking at standard poodles and Portuguese water dogs together. Okay. Okay, and yeah, it looks like so. And there was a comment here. There was a comment here about very interesting. The magenta dogs are all relatively non-shedding breeds, and totally, you know, that's it's one of these things where does it mean, for example, that the fact that they don't shed is somehow related to Addison's disease? Or were they just kind of bred together to be non-shedding? Were they just all bred to be non-shedding dogs? And this kind of also came along with it, right? It's sort of this concept called genetic hitchhiking that we don't really know whether it's just a correlative effect or a causative effect, if that makes sense. But you're certainly right. And I think for all of these kind of breeds that cluster together, right? And, you know, I'm just going to kind of trying to find my pugs here, right? But, you know, you could kind of make the same comment here. All of the blue dogs on the right, the Boxer, the Bulldogs, the Dog de Bordeaux, Boston Terrier, French Bulldogs, they cluster together and they all have smush mouths, right? So that's because they're highly genetic related and they were sort of bred to have the same kind of pattern or face or things like that. And that does lead to common problems in them, which we call brachycephalic upper airway syndrome, because of their kind of weird smush mouth conformation. So anyway, these are just some of the interesting things to notice when you look at the wheel of dog relatedness. But I want to get back to the sort of the clinical presentation here, and so we can kind of move on to the genetics. So I'm going to kind of advance here to the next slide. And so I wanted to sort of talk briefly about testing for Addison's disease. So how do we actually figure out if a dog does have Addison's disease? And this is stuff that I think many of you guys may know of. And so there's two general kinds of tests that works in, you know, 98% of cases for understanding Addison's disease. And the first test that we would commonly run is only a screening test. And this is a test we call a baseline cortisol test. It can be measured in-house with a SNAP test, which is sort of something called an ELISA, or oftentimes here, for example, we send it out to a reference laboratory. And what we're doing here is we're just measuring the amount of cortisol in a dog's blood at a random moment in time. And I apologize for anyone who's on the call from Europe. These numbers, the sort of units and the numbers will be a little bit different. These are sort of numbers that we use in the US, but greater than 2 micrograms per deciliter of cortisol is considered normal or healthy. And if it's below 2 micrograms per deciliter, we would say that the result is equivocal. And why is it equivocal? Well, so cortisol is normally secreted by the adrenal glands in different amounts at different times during the day. So cortisol, for example, spikes like first thing in the morning when we get out of bed. And people have done these studies in dogs to look at the timing in which cortisol gets released from the body. And so it's possible, if you just take a random sample of cortisol, that it could be low just because of the random fluctuations in cortisol throughout the day. And so if it's below 2, you can't really say whether or not a dog has Addison's disease. But if it's above 2, it's considered high enough that the dog, it actually rules out Addison's disease for sure. And so a lot of times if we have a dog that, we think might have Addison's or we're just not too sure, we'll oftentimes run just this baseline cortisol to like rule it out. But if the value comes in too low, if it comes in below 2, then you need to follow it up with, which again, in 98% of cases is a definitive test, and that's called an ACTH stimulation test. So what is this test? So this is a test where we inject into dogs a synthetic version of a hormone called ACTH, stands for adrenocorticotropin hormone, ACTH. And what we do is this hormone tells the adrenal glands, make more cortisol. So basically you take a dog's blood, you get a sample of blood, then you inject the cortisol, you wait an hour for the adrenal glands to do their thing, and then you draw blood again. So you essentially have 2 values. You have a before you gave it, and you have after you gave it. And in a healthy dog, those adrenal glands are going to be functioning normally. So the amount of cortisol they're going to give, the amount of cortisol in the blood should go up, right? Because that would happen in normally functioning adrenal gland. Whereas in a dog with Addison's disease, where that adrenal cortex isn't working, it should be flat. So you should be no changes in before and after you give the ACTH stimulation test. And this is actually some data from our own lab, just in terms of samples we've collected from genetic testing. And so what would you expect? So here you have on the left, you see a population of affected dogs. And on the right, you have a population of unaffected dogs. And then for each of these plots, the one on the left and the right, you see a before test and an after we give the ACTH. And you can see in the affected dogs, there's really no difference between the before and the after. So the adrenal gland did not secrete more cortisol after you gave it. Whereas in the unaffected dogs, you can see that all of the values here from before go up because the adrenal glands are doing their thing and making more cortisol. So this is how we can sort of definitively tell whether or not a dog, in fact, does have Addison's disease from doing the ACTH stim test, okay? And so that's really how we make the diagnosis. And as far as treatment, so treatment, we could have like a whole seminar on. I know there's sort of a lot of information out there about it, but I'm going to just talk very, very briefly about how we treat Addison's disease. And so one of the first things to know is, if you have a dog in Addisonian crisis, we need to worry about treating the shock. And that's for these 20 or 30% of dogs that come in the Addisonian crisis. If that's the case, it's going to require repeated fluid boluses. It's going to require treatment for the high potassium. These are often things that require being at a dedicated emergency facility or having a specialist around who knows how to deal with these life-threatening high levels of potassium, where you can have appropriate monitoring. But once you get over the shock, remember I mentioned that Addison's disease is the loss of these hormones, cortisol and aldosterone. We essentially need to replace these hormones through ongoing treatment. And typically the ongoing treatment is we replace aldosterone with percortin or Zycortol, which are these injections that dogs would be required to get. And these injections typically need to go every 25 to 28 days. It's something where every dog is different to determine the amount of aldosterone, how long it's going to stick around, et cetera, as well as cortisol. And typically in dogs, we treat the cortisol replacement with steroids or prednisone at essentially a physiologic dosing. So essentially just to replace what's being lost. Oftentimes steroids in canine medicine and human medicine can be used as an immunosuppressive dose, but for our replacement of endogenous or the normal cortisol that the body would synthesize, we use a much, much, much lower dose. Treatment, a couple things I'll also just mention about treatment and outcome. So once a dog's been diagnosed with Addison's disease, that dog needs both of these hormones to be replaced for the rest of its life, especially for the cortisol, because normally in the body, remember I talked about like stressful situations, which for the dog could be going to the vet or getting boarded. Normally a healthy dog would secrete more cortisol during those time periods. Oftentimes we tell owners to remember to double the dose of steroids if they're about to sort of put that dog in a stressful situation. And the other thing which has been shown is that if dogs are appropriately treated for Addison's disease with medication, regular monitoring, and treatment for the veterinarian, they will have a normal lifespan. So typically dogs don't die from Addison's disease once they've been treated and are sort of in a good spot. Sometimes we do lose dogs who are going through an Addisonian crisis or when it's not recognized. And there are even occasionally cases in humans where Addison's disease, kind of the great pretender, is not recognized, and humans can die of Addison's disease as well. So the question here is, can I speak to low dosage treatment? I'm going to say no, only because it's sort of a complicated subject, and it would sort of take us off the subject of what I want to kind of keep us on. I think there's sort of a lot of literature and a lot of kind of groups out there who are kind of dedicated to providing or sort of getting dogs on the lowest possible amount of prednisone replacement. And I think it's certainly like a reasonable thing to work toward, especially because there can be side effects from some of these steroids. But I think it's just important, I would say at this point, if you do have a dog with Addison's disease, to read up on this. I'd be happy to talk at some later date about sort of low dose steroid replacement therapy, but also just to kind of like work with your veterinarian for a dose of steroids that works for your dog. It's important to not let that steroid dose get too low, or it can sort of start to trigger many of the problems you would have in a dog that had no steroids in its body. So you do risk kind of putting your dog in some of these sort of Addisonian crisis-like situations if you don't give a sufficient dose of steroids. In any event, we'll kind of go from there. And it looks like, yes, Mary brought up there's an alternative drug that can be given that doesn't affect the ACTH thrust. That's a drug called dexamethasone. It's a drug that we typically give IV, although it can be given orally as well. Sometimes if we think a dog has Addison's disease, we can give the dexamethasone and it won't interfere with the ACTH stim. Normally, it's not one of those drugs that we use for ongoing treatment of Addison's disease. We normally use prednisone. In humans, they use a drug called hydrocortisone, but in any event, that's just sort of what works better in humans. But I want to sort of, only because we could spend forever talking about therapy for Addison's disease, and really my area of expertise is more around the dogs with the Addisonian crisis, since I'm a criticalist and I oftentimes really treat them when they're in the ICU and in the emergency room. I want to sort of change gears and talk a little bit about my research into this. And I'm just going to kind of quickly look through the chat to see if there was anything else. Addison's disease dogs are typically treated at lower doses than what they originally prescribed. Yes, and that is totally true. Like when a dog is just originally diagnosed with Addison's, especially if they're in the midst of an Addisonian crisis, they do need that higher dose of prednisone. But most veterinarians will work with owners to get the dogs down to a dose that works for that particular dog. It's important to remember that you need to give a dose that still is effective. So it can't just be, let's wave the bottle of prednisone under the dog's nose. You need to actually give a reasonable dose of the drug for it to be effective, because your dog really doesn't have any of it in its bloodstream if it's been diagnosed with Addison's disease. So I hesitate a little bit, when the dosages get so low, it sort of makes me a little bit leery. But obviously it's important to do what works for your dog and to work with your veterinarian. Can you talk about whether there is a typical age of onset? Yes. So great question. So typically the age of onset for Addison's disease is somewhere in the neighborhood of, we would think, 1 to 4 years, what you look at in the literature. it's pretty rare to see a dog over 9 or 10 develop Addison's disease. Now, the other thing which I will sort of say is that, we're focused here mostly in our research on the immune-mediated cause of Addison's disease, or the one that has a genetic predisposition. When you go back and think about the histology or the underlying disease of Addison's happens because the adrenal cortex is destroyed. And, I've seen cases, for example, where you have dogs with like lymphoma, which is a cancer of the lymphocytes, and the lymphoma can actually invade the adrenal glands and destroy it. And so you can actually have dogs that develop Addison's disease secondary to lymphoma, and that could happen at the age of 12, 13, 14, whatever. So sometimes you do see these later onset cases. It's not that they don't happen. It's just that it may be a different underlying cause when you see it in dogs at that age versus the kind of classic immune-mediated presentation. And so that's part of the reason in which we try to use dogs for our study that are sort of over 9 before we kind of call them controls or healthy dogs. Let's see, any other questions? Paolo didn't show until age 9, 10, but in retrospect, he was atypical that morphed into acute. Yeah, so again, totally, things can happen, that are outside the realm of what we typically see. But when you sort of look at the statistics, we typically think of Addison's as a disease of like earlier onset in life. That's at least that's the dogma that's out there. And I think that's supported by the samples that we've collected for our research when we look at the age of onset. But absolutely, there can be cases that sort of don't fit the typical bill. Are we going to discuss possible triggering events? Yes. So we are going to discuss possible triggering events as I kind of work through the research aspect of some of these things. Any other questions before I kind of move on? And I apologize. This is probably going to wind up running a little bit longer. So I would say like I have a little bit of time on the back end. I'll probably skip one aspect of the research thing, but I want to make sure I talk a little bit about the genetics. And then I do kind of want to get to this triggering event thing and that kind of thing a little bit. So I would just say, if you have to leave, feel free to leave. We'll leave this recorded, and people can kind of watch the end at another point. Okay. So in the absence of other questions, I'm going to move on and talk. Can vague symptoms go on for a long time? There was another question. Absolutely, those vague symptoms can go on for a long time. And I think oftentimes, it's because they're so vague and they're so waxing and waning that people just say, oh, my dog had a bad day today. Well, the next day they're fine. So we kind of just ignore it. So yeah, I mean, the vague symptoms can go on for months or years if they're not recognized. And sometimes, we see dogs, for example, that maybe the reason they don't get diagnosed with Addison's disease is because they have these vague signs that have persisted for such a long period of time. And sometimes that could be why we see some of these cases diagnosed late in life. It could also be, have a dog that like bounces from rescue organization to rescue organization and nobody sort of knows what's going on, or it didn't see a veterinarian who picked up on it, right? And so all of those things could be reasons why we sometimes see these cases diagnosed a little bit later. Okay, so I'm going to move on to research. And so as far as like my research goals here, right, I really have 3 research goals. And this is not something that, I want to accomplish in the next day, but this is, again, long-term, is number 1 is to predict dogs that are going to be able to develop Addison's disease. And this is something that's really important, I think, for the breeding community, for dogs to understand who's at high risk, right? And I think the one thing which I will sort of say about predicting dogs that are going to developing Addison's disease and the genetic work is that Addison's disease is what we call a complex genetic trait. And so what that means is it's not you have a mutation, you're definitely going to get Addison's disease. It's like, okay, maybe there's 5 or 6 things in a dog's genome that predisposes it to getting Addison's disease. And if you have 2 of those mutations, you have a 20% risk, whereas if you have 5 of those mutations, you have an 80% risk of developing the disease. And even if you have 6 of them, you only have a 90% risk of developing the disease. And there's going to be some kind of combination of genetic mutations plus some kind of a predisposing trigger. And so that's why it's never going to be one of those diseases where we're probably able to predict with 100% accuracy, this dog's going to get it, this dog's not. It's going to be that these dogs are at higher risk, these dogs are at lower risk. But then still there's going to need to be some kind of a trigger, some sort of a thing that actually allows the Addison disease to happen. And as I go through the research here, we'll maybe talk about some of those things as well. So anyway, first goal, to predict dogs that are going to develop Addison's, but I don't ever expect it to be 100% ability to predict it, like you could with the mutation that was a simple recessive or a dominant trait. So the second goal is really to develop new ways of preventing and treating Addison's disease. And so, interestingly, like especially if you think about to really beyond the typical treatments that we use, which are hormone replacement, if we can predict, for example, that this dog is at high risk of getting Addison's disease, is there a way to actually prevent that from happening? And so there's a lot out there in the literature, particularly when you look at like mice and humans, where people are gravitating toward ways of manipulating the immune system to not allow the onset of autoimmune diseases. And so it's a little bit cutting edge, but that would be the goal once you can predict the dogs that can develop it. And then finally, to try to kind of understand some of these environmental triggers, because like I said, it's not going to be solely a genetic problem. Okay, and so our approach to look at both the genetics, the immunology, and some of these environmental effects. Okay. So let's sort of talk a little bit about the genetics and kind of where we are with what I've gone through in the past, what we've sort of found so far in Portuguese water dogs, and kind of where we're going. Okay, so here's how a typical genetics roadmap to trying to kind of understand inherited diseases in animals. So initially, we'll start oftentimes with something called a genome-wide association study. And I'm going to talk about what that is in just a second here. But this is sort of a marker-based approach to try to find regions of the genome. So the dog genome is about 3 billion bases in size, to try to understand those regions of the genome that are associated with the disease. Once we kind of find those regions, sometimes we'll do something called fine mapping, which is really to look at that region in careful detail and look at the particular regions, some smaller regions that might be associated with the disease. We'll then either focus on sequencing just that region, or nowadays it's actually even cheaper just to sequence the entire genome of the dog to look at every base within that region, and then to try to find actual causative mutations using that whole genome sequencing. We'll oftentimes then try to validate those findings in larger populations, and then do these kind of functional studies to try to understand, okay, what do these mutations actually do in the body? And so the classical approach, which I mentioned earlier, relies on something called a genome-wide association study. And the reason I go through all this detail is I'm going to show you some of the early findings that we had in standard poodles, and also some early findings that we have in Portuguese water dogs, so that you can understand, what's the basis of a GWAS and how you interpret the data that you're going to look at. So as we all sort of know, that the way in which genes and disease are inherited are through DNA. DNA is composed of, in the dog, about 3 billion bases. And so a base is one of these, typically is, there's 4 different bases that make up DNA. There's a T, which is thymidine, a G, which is guanine, an A, which is adenine, and a C, which is cytosine. And so this is just a fragment of, let's say, 10 bases out of an entire genome of about 3 billion bases. And throughout the genome of any animal, of any person, we have these variants, which are called single nucleotide polymorphisms, or SNPs, we oftentimes refer to them as. And so what this means is that even though, let's say my DNA, right, looks like it says on the top, where I have T, G, A, G, A, blah, blah, blah. In some other humans, let's say, I'm just looking at Mary here. In Mary's DNA, there's a substitution of, instead of having a G at this position, she has a C here. And in Mary's DNA, instead of having an A here, she has a G here. And instead of having a C here, she has a T here. And this is what I would just think of as like benign, inconsequential variation that exists throughout the genome. And these are called SNPs. And what we do in the dog is like we have these SNP chips that actually allow us to genotype. So to determine in a particular dog, is there a G here or a C here? Is there an A here or a G here? To look at about 700,000 of these SNPs all at the same time in one array that cost about $100 to run. And we essentially look at all of these different 700,000 SNPs at a single time. And what we look for is we look for a marker that is associated with the disease. So if you look at this particular SNP, you could say all of these dogs here, right, they're smiling. And you can see they all have this green dot. So they all have this G, right? So these are your healthy dogs. Every single dog has the G. And here you have another population where all of these dogs are frowning, but they all have the C, right? So here you have all of the healthy dogs have a G, all of the sad dogs have a C. And so maybe if we were to sort of look at this in a larger population, that this particular single nucleotide polymorphism is associated with the disease that we're interested in. And that's essentially the basis for a genome-wide association study, just across many, many larger populations of dogs and many, many different more SNPs. So the way that I sort of started my foray into Addison's disease, as many of you guys know, I started looking at Addison's disease in standard poodles. And we went through kind of large sample collection efforts to collect data from standard poodles, where we sort of looked and made sure they had an abnormal ACTH stim test, they had to have an abnormal sodium-potassium ratio, they had to have consistent clinical signs, and we called these affected dogs. And then we also had a sort of a similar population of unaffected dogs, where we had to sort of rule out Addison's disease. We had to make sure they were at least 9 years of age. And we collected through the course of this research, and we've actually done more since I put together this figure, 77 affected standard poodles and 103 unaffected. And this is the result that we had from this genome-wide association study. And this is something that's been published, but I want to sort of explain to you what we're looking at here. So in this figure, this is a figure called a Manhattan plot. You see on the x-axis all of the different dog chromosomes. So the dog has 39 chromosomes, including the X chromosome, and you see them 1, 2, 3, 4, 5 here. And on the y-axis, we see something called a p-value, which is sort of a measure of statistical significance. And each dot represents a single SNP, like that I showed you on the previous page. And the height of the SNP represents how associated that SNP is with Addison's disease. So if it's low, that means it's not very associated with standard poodles with Addison's. And if it's high, it means it's very associated with standard poodles with Addison's. And this blue line here is sort of a line that we typically use to represent statistical significance. And what you can just kind of clearly see looking at this figure is there's not a single SNP that reaches the line of statistical significance. So after basically working really hard, get lots of standard poodles to make sure they're as unrelated as possible, to make sure that the cases are cases, the controls are the controls, looking at the pedigrees, we do all this work, and we essentially find that there's not a single nucleotide polymorphism associated with Addison's disease in the breed. So kind of a little bit of a frustrating result. And this is the point at which we kind of needed to step back and see why would we have no significant hits in a genome-wide association study. And there's a number of different reasons. I would say that these are all hypotheses. We don't know the answer for sure at this point, but a number of different things you could imagine. So number one is that it's possible, and it's definitely consistent with the fact that Addison's disease is a polygenic trait, meaning that there's multiple different single nucleotide polymorphisms associated with Addison's. It could be different in some subpopulations of poodles versus others. And because each one just has a very, very small effect size, you just wind up needing a much, much larger population in order to sort of see a significant hit. And so that would be kind of classic for a polygenic trait. Another sort of thing, which is kind of consistent, is something we can say in genetics called an incomplete penetrance, meaning that not all animals with the variant actually develop the disease. And so this is really where more you start to think of, okay, you can have some animals that have mutations, but never develop the disease, for example, because they never had the environmental hit that triggered the disease. And so that can mean, if you either a polygenic trait or incomplete penetrance, that you just need way more dogs than you expected in order to actually develop the disease, in order to sort of get a significant hit on a genome-wide association study. It's also possible that the variation, so there are certain variation in the genome, certain genes that are important in autoimmune diseases called major histocompatibility complex genes. There are also structural variants in the genome, which are things like chromosomal rearrangements. These are not really well captured on genotyping arrays, so it's possible there's something going on that we're just missing. And the other sort of possibility, and this is something that Mary was had mentioned to me just as we were kind of getting started, is it's possible that to some extent, I'm not saying entirely, but the predisposition to developing Addison's disease is actually kind of fixed within standard poodles, meaning that to some extent, every standard poodle is just as predisposed to getting Addison's disease as the next. But the important thing to understand is to think about what is the environmental trigger that's making them predisposed. So maybe there's something, that has gotten hijacked along with the curly coat gene in standard poodles that makes all poodles predisposed to developing Addison's, but it's only the ones that have an environmental hit that get it. And it's also a possibility that there's some combination of this, right? That like 40% of the risk, and I'm making up these numbers, right? 40% of the risk is fixed and 60% of the risk is variable. And of that variable portion of the risk, there's 5 different mutations. And depending upon how many of those 5 you have are going to determine how likely you are to get Addison's disease. So any combination of all of these things are really possibles. But this just goes to show you how hard it is to try to understand and dissect these complex traits. There's a couple of questions here. How do you determine the line of statistical significance? Is it a standard figure? So yeah, so the answer it's kind of standard. So the line of statistical significance is highly dependent upon the number of statistical tests that you run. We use something called the Bonferroni cutoff. And so what that does is it means that the level of statistical significance is 1 over the number of tests that you run. Typically, the number it's around 1 times 10 to the minus 7, 1 times 10 to the minus 8 would be this line of statistical significance that we use, but it really depends upon the number of tests, but it's sort of a well-defined statistical metric. And is it possible to fix in Portuguese? Mary had a question in the chat. Is it possible that it's fixed in Portuguese water dogs? 100%, right? It's possible that there's some degree of fixation, complete fixation, partial fixation. No, we don't know. But from the typical genetic test that we run, we can really only suss out the portion that is not fixed. Because if it's fixed, it's going to kind of be the same in everything. Now, there are some kind of newer genetic methods that are out there that we're going to be employing in Portuguese water dogs that try to get at understanding what are the fixed regions of the genome, but it can be harder to determine, to necessarily associate those with Addison's disease. But so it's possible we can get at it. It's sort of definitely on our radar screen. And one of those things that, as a matter of fact, Lizzie, who's one of my grad student who's working on this disease, and I were just chatting about last week at our weekly call. Okay, so I want to sort of get on to talking a little bit about what do you do with something like this? And so our next steps really in standard poodles, collect additional samples, minimize more relatedness, add more dogs to evaluate some of these genes that we don't capture on typical arrays. There's these major histocompatibility complex genes. We're also trying to employ some new genotyping technologies so we capture more of the variation in the genome. This is actually something we're doing in Portuguese water dogs. But the other thing really, when we think about Addison's disease as a whole, is to kind of add breed diversity, right? So I showed you guys a couple of slides ago that poodles and Portuguese water dogs, highly related. Our hypothesis is that they're going to share the same genetic makeup. And so by studying Addison's disease in both poodles and Portuguese water dogs, we greatly increase the likelihood of us being able to find something because the 2 breeds are, genetically different, but also genetically close. So we have an active study that was funded by the Portuguese Water Dog Foundation in 2000, kicked off in May 2001. And our goal is really to try to understand the genetic basis of the disease in Portuguese water dogs. This is a picture here on the right of Earl, who's the Portuguese water dog owned by my other grad student. Her name's Amy. You can see the dog has a University of Minnesota thing on its collar. But anyway, we're basically trying to use some of these cutting edge genetic techniques, some newer technologies for doing SNP genotyping, whole genome sequencing, and also direct sequencing of these key immune system genes to try to understand what's going on in Portuguese water dogs, with the idea of looking at it obviously in Portuguese water dogs alone, but also trying to be able to aggregate the data that we have together to try to understand it as a unit, with the hope that understanding it in one breed will help us understand what's going on in the other and in the disease as a whole. Okay, so in terms of what we have found to date, a lot of our effort to date has been focused on sample collection, and this is just an update on numbers for you. And so one other thing, just to kind of, we've also started a collaboration as part of this with a researcher named Anita Oberbauer. She's at University of California, Davis. She actually had spent some time trying to understand the genetics of Addisons and Portuguese water dogs some years ago, and she's kindly given us access to all of her samples for this study. And so in combination of your guys' efforts, Lizzie's efforts, Lizzie is the grad student who's working on this primarily, Dr. Oberbauer, to date, we've collected 130 samples from 131 affected dogs and 167 unaffected dogs. Now, if you look at this next column over here in terms of qualifying based upon relatedness, you can see that these numbers are dramatically smaller. And one of the things that's important to do in genetic studies is, dog breeds look the same because they're all highly related, but that relatedness can also make genetic studies a little bit more difficult to do. And so when we include dogs in our initial screen, we like them to be unrelated at least the grandparent level, hopefully the great-grandparent level. And so many of the samples, because they have come primarily from the United States and Canada, are not qualifying for the study, at least in our initial screens. We can certainly use them later. We have about 41 affected and 43 unaffected. We have a number of different samples that are pending, so 7 affected and 39 unaffected. These dogs we've already screened to be unrelated, and these are samples that we're working on getting from overseas, and so that's a work in active progress. But our goal is to get about 100 unrelated affected and 100 unrelated unaffected dogs. And so we'll kind of see where that goes and sort of adjust that number as we move on with the study here. But so far, because we wanted to try to dig into this data, as soon as possible, we've done basically using a subset of these unrelated samples, these 41 and 43 dogs. We did run an initial genome-wide association study. You can see there's 544,000 markers represented here. This includes 28 affected Portuguese water dogs and 35 unaffected. We use this kind of new technology called low-pass whole genome sequencing. But this is the same kind of Manhattan plot that I showed you for the standard poodles, but this has a much smaller numbers of Portuguese water dogs. And here, this line of statistical significance is in gray. You can see that, there's some kind of maybe interesting peaks here on chromosome 23. Maybe there's an interesting peak here on chromosome 30, but nothing that gets us super excited yet, because nothing really crosses that threshold for statistical significance. All of that said, when we add more dogs to the study, so we've already collected unrelated dogs that we haven't yet genotyped, we're collecting more. We're probably in the next month or two going to submit another batch for low-pass whole genome sequencing. Our hope is that this numbers, or some of these regions of the genome, will start to reach statistical significance, at which point we can kind of really dig into them. So, but this is just kind of where we are to date. Wanted to make sure everyone's seen at least some of the progress that we've had so far. Another thing that complements this, I mentioned that we sort of have some suspicions that Addison's disease is an autoimmune disease. So it's really important in autoimmune disease to interrogate these genes called major histocompatibility complex genes. This is a little cartoon of a major histocompatibility complex protein. And this is just what the protein looks like, but these proteins are really, really important in the immune system, because within this little cup here that you see, that I've had with my cursor, the immune system presents peptides or different substances to itself. And so if the immune system, for example, presents to itself a piece, for example, of the adrenal gland, it could set the immune system off to go ahead and attack the adrenal gland. And these MHC genes are also like among the most variable genes in the body that are really important for immunologic diversity and the body's ability, to respond to multiple pathogens. And so one of the things that we want to do is we want to be able to genotype all of these genes in Portuguese water dogs and understand if some version of these genes is associated with Addison's disease. And one of our interesting findings so far is we actually have found one kind of version of these major histocompatibility complex genes that seems to be associated with Addison's disease in Portuguese water dogs. If you look across the Portuguese water dog genes, there's probably about 7 or 8 different versions of these genes in Portuguese water dogs. And we found one version of these genes that you can sort of see here by these statistics. About 55 or 60% of cases have this version of the gene, whereas about, under a quarter of unaffected dogs have that gene. And so this is pretty significant at a p-value of 1 × 10⁻⁵. And so as we kind of continue to genotype more Portuguese water dogs, we'll see how these statistics hold up. But this would be pretty exciting if we're able to sort of find this risk haplotype. But keep in mind that a risk haplotype doesn't mean you're definitely going to get it, right? You can have the haplotype and still be healthy, and you don't have to have the haplotype to develop the disease. But this is just one of these things that we oftentimes look at from when we try to understand autoimmune diseases. Question. So yes, so there was a question here, or I'm just sort of looking at some of these questions here. One question that was asked around, is there any sort of breakdown among curly, wavy, and all that kind of stuff? So yes, so we've actually, all of the dogs that have been enrolled in the study, we're definitely collecting coat color and coat style with all of the other information we collect, as we're also asking for like weight and height, because we have some preliminary data to show that there's some association with weight and height and Addison's disease. And so I don't think we've run the statistics on that yet, but we're certainly preparing to be able to run those statistics. And even though, while I think it's unlikely that things like coat style or coat color are directly causative of Addison's disease, it's possible that some of the genes that are associated with Addison's disease kind of hitchhike along with those things. So we might see, for example, that there's a higher incidence of Addison's, for example, in curly coated, versus wavy coated. And I don't know the answer to that question, but we're collecting the data to be able to understand things, associations with like sex and that kind of thing. And so all that research, that analysis will be done. And the other thing is, to the best of, there's been some kind of reports in the literature, about association with sex, although the biggest study from the one from Sweden showed that there was no association with sex, but I think it's still an open question. Okay. So let me sort of go on and we'll say, like, how can you help here? So essentially, really what we're looking for is any dogs that are unrelated to the population that we already have. So we're looking for, if you know, or you have, or you know of a Portuguese water dog with Addison's disease on the left, they can be any age. We want only the typical Addison's dogs where there's a pedigree available, so we can evaluate the relatedness of the dog to the population that we have. If the dog is sufficiently unrelated, we would love to get a blood sample for our study. Typically, to get these blood samples, we work with your veterinarian to draw the blood if they're based in the US and Canada. Overseas, we're working with cheek swabs instead, just because it can be a little bit of a pain to get blood from overseas. And we do have a particular need for non-US dogs, because as we try to minimize relatedness, being able to get samples from Australia, Norway, wherever, is going to be helpful for us in minimizing the relatedness to the population that we already have. We also always want to balance the cases with the controls. So for example, we don't want all of our cases to be from the United States and all of our controls to be from Sweden, that kind of thing. That would create very skewed populations that would impair our ability to look at the genetic data. As far as, and so for for Portuguese water dogs without Addison's disease, we'd like them to be at least 9 years of age with no concurrent other autoimmune disorders and have a pedigree available. And again, a particular need for non-US dogs. If you do know a dog who might qualify for the study, we would ask you to kind of please reach out to us. At this address, pwdstudy@umn.edu, and let us know what you that you'd like to participate. And Lizzie, who checks that email regularly, will kind of follow up and work with you to get a blood sample if you're in the US or Canada, or a cheek swab if you're overseas. And the study will pay for all of that, as much of that as possible. So. Great. So that's kind of all I wanted to say about the genetics. I know we're kind of way over time here. So question came up, are we recruiting directly through? Yes, definitely. We're recruiting through PWD groups, through Facebook. We're also recruiting samples through the CARE website. Maybe all of you guys would know that if you have a dog with Addison's disease, but I think it stands for Canine Addison's Resources and Education. It's kind of this group that's sort of born out of Facebook. They have great resources for dogs with Addison's disease. We've done webinars for them. Obviously, it's beyond Portuguese water dogs, but sometimes owners who are sort of not part of the kind of Portuguese water dog breeding community necessarily, they just have a pet Portuguese water dog, will kind of come in through more of the Addison's related groups as well. But yes, we are searching all online channels that we possibly can. I think if you know of a channel that you an online group or something like that you think we have not yet targeted, if you just let Lizzie or send an email to PWD study, a lot of times we'll talk to the moderator and ask them, can we post about the study? And we'll just post a blurb or a link to the web page about the study, or just a link to the email address. So yeah, we're doing our best to kind of reach out. Really the difficulty is with relatedness. As I'm sure many of you guys know, the Portuguese water dogs are a small breeding population. And okay, so it looks like someone reached out a couple of days ago. We'll work out. We'll make sure we check that email. I think as long as you send it to PWD study at UMN. I don't know that we don't necessarily check that email every minute, but we'll definitely get back to you, I think, within a week on those. Okay, so just because we're a way over time here, and I actually have some other things to do, I'm going to sort of quickly, the one thing which I did sort of want to talk to you guys about, and this is probably going to be not so much a can of worms, but it's going to engender a little bit of a discussion. When one of the questions that can come up was around environmental triggers for Addison's disease. I'm going to talk to you guys, for those of you who want to stay on, about some findings that we've had from some of the other work that we've done. So I mentioned sort of beyond genetics, we sort of have a bunch of different studies looking at the immunology of Addison's disease, and I'm going to kind of skip all this stuff and sort of go and talk a little bit about some of these environmental triggers. And then we'll kind of end the discussion here, and I'm sure this will generate a bunch of questions as well once I talk to you about these results. Okay, so as I mentioned, Addison's disease is a complex disease. It's going to be polygenic, many genes. There may be some degree to which the disease is fixed within the breed, but it's not totally a genetic disease, meaning there's something else that might take a dog that has all the predisposing genetic risk factors to either get the disease or not get the disease. And so one of my goals in research is to try to suss out what could be some of these environmental triggers that lead a dog to develop Addison's disease beyond just the genetics. And I will say this is a challenging area of research, whether you look at human disease or canine disease to try to figure out what environmental triggers cause a disease. is really, really hard and challenging. The best example that's out there, and I that just to sort of bring up is that, for example, there have been really clear research that's associated, for example, cigarette smoking with certain autoimmune diseases like rheumatoid arthritis. And it's kind of gone down to the molecular level. So cigarette smoke can cause something called citrullination of proteins. And some of those citrulinated proteins can sort of trigger the immune system in a way that non-citrulinated proteins can. And so this is kind of the classic example of how some environmental triggers can lead to the onset of autoimmune diseases. And so, but getting to that level of granularity, like I give that example, but that's probably maybe the only really, really well characterized example of how the environment can cause autoimmune diseases. And, I think people really haven't been able to draw such great links, but it's kind of an emerging area of research. And so it's one of these things that is being actively looked at in not only the dog world, but also in the human world. And so one of the ways in which we've tried at a first brush to try to understand what's triggered Addison's disease is to try to do an epidemiology survey. And so some of you guys may have been involved in this. So another one of my grad students, Amy Treefel, as part of her PhD, put together, it was maybe a 10 or 15 minute survey that we had out in the world. I think we ended it maybe a year or so ago, and it was open for about a year. And it was open to try to understand some of the demographic characteristics, some of the exposures in the indoor environment and the outdoor environment that animals are exposed to. And this study was open to owners of both standard poodles and Portuguese water dogs living in the US and Canada. And the reason we wound up restricting it to the US and Canada is really because one of the other things that we had wanted to do is to take the location where standard poodles and Portuguese water dogs live, like down to the zip code, and overlay certain environmental databases that are available from, for example, the US Environmental Protection Agency and things like that, to see if there's any association, for example, in like proximity to Superfund sites or proximity to contaminated groundwater or proximity to poor air quality and Addison disease. So we sort of took all these survey responses, we took the location where all of these things happened, and we basically wanted to sort of overlay the data and see if there was kind of any association. Okay. Great. And so we did this survey and recently submitted a manuscript around this, but I kind of want to show you some of these initial findings here. And so what we did is we ran this survey, and I'll kind of walk you through this. So we had a total of 6,286 responses to the survey. So this was a pretty good response. I will say that the majority of the responses were owners of standard poodles. I think that's just a function of the fact that standard poodles are more popular than Portuguese water dogs. We removed a number of these surveys, so 7 dogs were ineligible for whatever reason, 6 dogs people tried to submit at the same time, 1,226 people didn't complete the survey, so it was less than 20% complete, we threw it out. And so we wound up including a total of 5,047 cases in the initial response. And of 547 surveys, and of those 5,000 surveys, 358 of them were cases and 4,689 of them were controls. And the reason why you just see an unbalanced population between cases and controls is this was just a survey that we submitted to everyone. And as you can imagine, there's a much, much greater population of healthy dogs than affected dogs out in the general population. Now, what I'm going to show you here on the next page is something that some of you may find interesting, surprising, controversial, but I'm going to sort of go here on the next page and show you. And so probably the most significant finding from the study, and I'm not going to go through all the different questions we asked things about, like, flea and tick products and medications and campfires and bodies of water and sort of candles and cleaning products and pesticides. All sorts of questions were asked, but the one that was kind of far and away had the most significant association with Addison's disease was spay or neuter status. And this was something that was like a little bit kind of maybe unanticipated that we found. But what we did find in the study was that particularly for neutered male dogs, had a 6-fold increased risk of developing Addison's disease compared to unneutered male dogs. And spayed females had a 2.5-fold increased risk of developing Addison's disease compared to intact female dogs. And you can see there's like a little bit of a range here, right? So, if it's a 6-fold risk is what the odds ratio was, there's a range of 2.6 to 2.5 to 14-fold. And for females, it was, 1.4 to 4.5. But, the p-values are kind of highly significant for being associated with dogs with Addison's disease. And so we did a number of different other adjustments to the study here, for example. And if I go back to the next page, so once we sort of started with these 5,000 dogs, one of the first things that we found is that a lot of the dogs who responded to our survey actually had young, healthy dogs. They might have been like under 9 months. So essentially, we asked ourselves the question, well, maybe they were just, there were fewer healthy dogs that were just too young to actually get spayed or neutered. So we wound up kind of restricting our analysis to a population of 330 cases and 2,700 controls that were at least 25 months of age, so at least 2 years of age at the time of the survey, where they would have at least been eligible to be spayed or neutered. And still, this figure of being a 6-fold increased risk of developing Addison's if you're male and a 2.5-fold increased developing Addison's if you're female, all of those ratios still held up. We did another kind of follow-up survey to kind of look at this. And so one question that we asked ourselves is, well, could it just be that the dogs with Addison's disease, owners found out that they got Addison's disease and then decided to spay or neuter them? So we wanted to get a sense for, did the spay or neuter procedure happen before or after they were diagnosed? And the quick summary of this answer is that at least 80% of the affected dogs were altered before they were diagnosed with Addison's disease. So people were not generally spaying or neutering their dogs because they found out they had Addison's disease, but all of this spaying or neutering happened, most of it happened before the diagnosis. And this is just a plot showing the distribution. If this red line here is the time at which a dog was diagnosed with Addison's disease, what does the distribution look like of when the spay or neuter happened prior to diagnosis? So, for example, there are some dogs that were spayed or neutered 120 months, so about 5 years, or maybe even more, 10 years before they developed Addison's disease. But quite a number of dogs were spayed or neutered about 2 years, 20 months before they developed Addison's disease. So, kind of quite some time. And so, interestingly, this was really a kind of interesting finding, one that we didn't necessarily expect to find, and one that's for sure going to inform the way in which we approach the genetic aspect of this thing. And so what I just kind of want to emphasize is what some of the takeaways here are from the survey, but it does suggest a possible role for spay and neuter in the onset of developing Addison's disease, where there would be a higher risk for males versus females. If you dig into the literature, this finding is actually supported by 2 other studies in the literature. One of these studies came out of the UK, and another study came out of UC Davis. They looked at different populations of animals. So, for example, none of them were as breed restricted as this particular study. But it is sort of supported by 2 other studies in literature. it certainly doesn't explain the entire story of Addison's disease, because certainly we had some dogs that were spayed or neutered after they developed Addison's disease. And the mechanism behind what's going on here is not really clear. So there have been lots of studies out there showing important effects of hormones on the immune system. So the immune system, hormones can modulate how the immune system works. It's also certainly possible that gonadal hormones, so hormones from the uterus, from the ovaries, and from the testicles, can directly impact the adrenal glands. There's certainly an interplay between the adrenal glands and the sex organs. And then importantly to say is that, this finding really just shows a correlation, doesn't necessarily show a causation, but it definitely gives us an area to think about investigating from more of a mechanistic perspective. But from a genetics perspective, this kind of a finding will definitely inform the way in which we approach our genetics research by taking spay or neuter status, for example, into account when we do our genetic studies, and or by sort of subdividing our population into, let's look at just a population of spayed or neutered dogs, and let's look at a population of intact neutered dogs. So it's for sure, it's something to kind of look at. And I think the other thing, which I just kind of last want to say is, this doesn't mean don't spay or neuter your dog, or don't recommend people to not spay or neuter your dog because you're going to get Addison's disease. That's not at all what we're saying. All we're saying is that based upon this initial survey, there appears to be some sort of an increased risk of developing the disease if you are spayed or neutered. Exactly what that risk is sort of an open question. Okay, so there's a couple of other things that kind of came up. It looks like there was a question around relatedness. And so, yes, so there's sort of a question around, it's a good idea to have for 3 generations. The other thing I will say is, so in the way when we do the statistical calculations, even though we only include dogs that are unrelated back 3 generations, still the dogs are going to be somewhat related. And the way in which we do the genome-wide association studies, we actually create what's called a relatedness matrix using the program. And this relatedness matrix gets used as like a covariate in the statistical model. So even though we'll try to eliminate dogs that are related back 3 generations, we also do take into account kind of the remaining underlying cryptic relatedness in our genetic study. So it's a great point. And all you can try to do is minimize it as best you possibly can. There's no 100% of getting away from relatedness when you do genome-wide association studies in dogs. Looks like someone commented, a GIS background can help with the mapping. So we've actually already done the mapping studies. And when you look at the association, they're really, at the first pass, there really was no association with Addison's disease in terms of like living in an area where there's poor air quality, or living close to a brownfield site, or living close to contaminated water or soil or something like that. So we've already done that analysis, and that will be come out in the study. I think I was just trying to highlight what was the most statistically significant in the environmental study, which is the spay-neuter thing. All of the other things that we were kind of thinking might have an effect, like, like the spot-on flea or tick products, or, the type of diet, and, all those kinds of things really did not have an effect. One question which may be lurking out there in the background is, did we sort of look at vaccines and timing of Addison's disease? We did not attempt to do that in this survey. I think trying to get at that information is maybe valuable to do at some point, but it's really hard to get that information from a survey, and probably requires more of an in-depth looking at actual medical records rather than trying to get that information through a survey. Could you attribute to the stress of the surgery or missing hormones? That also came up. And so I think that's a good point, although I think we would have, and that was one of the other reasons why we wanted to look at the timing of the onset of Addison's disease relative to the spay or neuter time. I think if, your average dog was spayed or neutered 24 months prior to developing Addison's disease, like the imminent stress of the spay-neuter procedure would just be too far away to sort of think of it as a causative effect. But it was certainly one of those things that we considered in the study. Okay, so there's a new message here. Yep. So that's also a really good point. And so it looks like Angela brought interesting. The rate of Addison is much higher in the Swedish study then. So it's actually kind of interesting. So when you look at the overall population incidence, so that the information that I gave you from that survey from Sweden actually was about the relative risk of Addison's disease. So the risk of a Portuguese water dog in Sweden developing Addison's disease relative to an average dog in Sweden. So that's what that study was showing. But elsewhere in that study, they actually did report on the overall incidence of Addison's disease. And one kind of really interesting thing, there's no great epidemiologic studies of Addison's disease in the United States, but a couple of studies have looked at it briefly and sort of said maybe, up to 7% of standard poodles or up to 3 to 5% of Portuguese water dogs have Addison's disease. The overall incidence in Sweden is a lot lower for Addison's disease. And in Sweden and Europe, spaying and neutering is much, much less common. So it would make sense that if there's a role for the sex hormones in the development of Addison's disease, that the incidence of Addison's disease would be lower in a country that doesn't as commonly spay or neuter their animals, as it would be higher in a country like the United States, where spaying and neutering is much, much more common. Any correlation, looks like pregnancy is a trigger. So people do think of pregnancy as a trigger. It's not something that we looked at or not something that we tried to, I have any data on. But certainly pregnancy is one of those things that can trigger a stress response. And I feel like I've heard of a number of different cases kind of anecdotally where, oh, my dog developed Addison's disease when she was pregnant, or we just diagnosed it after she sort of gave birth and sort of didn't do a great job in sort of like nursing or things like that. So there was really no correlation that we really tried to investigate whether or not pregnancy was related to it. So don't have a great answer to that question. But it's certainly a good thing. It's a good thing to think about. Although I do think that, the vast majority of owners who responded to the survey, again, most of these dogs are going to have been spayed or neutered and would be part of the general pet population, since that was the sample that we kind of targeted in broadcasting about this study. Okay, so that's kind of really all I have. Just kind of a reminder to throw up this slide here of how you can help. Really what we need is sort of getting back to this, trying to understand, trying to get at the genetics to enroll more unrelated dogs. If you think you have a dog that's going to qualify, please email us. And I kind of just want to thank everyone, the AKC, the Poodle Club of America, English Cocker Spub, Portuguese Water Dog Foundation, the PWD Club of the Twin Cities, Portuguese Water Dog Club in general, the NIH, everyone in my lab, and some of my mentors who've been helpful for this research, just to kind of thank everyone who's been involved in this research. And if there's any more questions, I am happy to take them either by raising your hand or the chat. Otherwise, we can finish up. And again, I apologize, this went about 40 minutes longer than I thought. These charts be available for download? No, but the paper is sort of in the process of being reviewed, and some of those statistics will be available, but the slides themselves, I was not planning on making available. But you can certainly watch the presentation, that'll be available. Any other questions? Okay, well, thank you guys for coming and for listening, and for the 37 of you who tolerated this talk to the end. I hope you found it informative. And please do email me or email the study if you have any additional questions. And I just want to say thank you again for your support, not only to the Portuguese Water Dog Foundation, who funded this, to the Portuguese Water Dog Club, who's helped a lot with sample recruitment, to all of the owners, breeders who've helped recruit samples. You know, we could not do this research without you. And we really, really appreciate your help in making all of this possible. And also, you know, thanks to Lizzie, who's done the bulk of this work on the ground here. I don't know if she's still on the call. And also, you know, thanks to Janet and Mary, Janet from the foundation and Mary from the club and Cheryl from the club, who've been some of the folks who I've been in contact with. Questioning about how will you get access to the recording? I think it's going to be posted on the foundation website, if not other places. I guess some email will probably come out about that, but I think if you look at the foundation website, it'll be available as soon as at some point when they get it uploaded. So, okay. No, it says recording will be available on the foundation website research project, and they'll post it on Facebook as well. So. Great, absolutely. Thank you guys for everything. And yeah, feel free to drop off. I'm happy to hang around. If there's any other questions, I'll stay on for just a couple seconds. So I'll stop my screen sharing. Oh. Lizzie, you stuck around.
Of course. Very good talk, as usual. Good job.
Thank you. Anything Mary or Janet, do you want to talk about, or was that good for you guys? Or
I think it was great. I really appreciate it.
Okay.
I love the new
get this recording up. I'll get it to our recording helper.
Okay,
Should be up there in the next couple of days. I'll let you guys know when it's up.
Okay, that sounds good.
Yes, I even learned some things I never knew, and I thought I knew everything. No, about Addison. When I had my Addisons, I thought I learned. You know, I had a really good grip. I was just even the biological representations. They were very informative. I think it was. I can't thank you enough for doing this.
Good. Yeah, I'm happy to do this. Like I think education and outreach is like super important. You know, and I'm happy to kind of give back however I can. So, you know, thank you guys for helping shepherd this through. And, you know, if you have any other comments or concerns or whatever, just let me know. But other than that, I'm going to kind of go take my dog for a walk because he's like sitting here at my desk needing to.
So all right, mine's underneath my desk too, so she's ready to go out.
Okay. Well, thank you everyone for coming. We really appreciate it. And Lizzie, I'll catch up with you tomorrow morning. But if you need anything, let me know. Okay.
Thank you so much, Steve.
Thank you guys. Talk to you later.
Bye-bye.
Bye.
I was Elizabeth.